more than that. So please represent India.
So we are live.
Okay. So I think I can see Dr. Vikram Raha also. Dr. Vikram, in what capacity you are with in this faculty panel? Harshit, should we start now?
Yes, sir. We are live. We are live, sir. Okay, start.
Okay. Welcome, friends. Greetings from American College of Physicians. I, Dr. Anuj Maheshwari, Governor, American College of Physicians, India chapter, welcome you all in this very innovatively thought nutrition symposia, which is the second nutrition symposia in the month of September, which is dedicated for the nutrition and science behind the nutrition. And today we are having Dr. R.B. Singh as our very learned moderator, who is from Simsong Institute, Poland, and he has been the eminent researchers with 550 research papers, nutrition, cardiology, and metabolic science. At the same time, he is the editor of the World Heart Journal, which is being published from New York and USA. And he has been been author of the 7 books which has been published by the American publishers. And very significantly to be noticed, he has earned 18,000 citations on his research work and 12 awards. So with this rich biodata, which is CV, I welcome Dr. R.B. Singh. Dr. Narsingh Verma, who is a close associate of me, also the secretary of American College of Physicians India chapter. He is the convener of this program. And all the topics which are being discussed in this program are the brainchild of Professor Narsingh Verma. Professor Narsingh Verma is president of the International Society of the Medical Nutrition. Apart from that, he is head of department of the family medicine at King George Medical University. He is professor of physiology in the same university. Apart from this, he is having many accolades and many awards which has been awarded to him. To welcome Professor Narsingh Verma.
Thank you.
We are having 3 very eminent speakers with us, Dr. Debashish Bakshi, Dr. Shipra Bhardwaj, Dr. Arun Kedia. And today's topics are very much different than routine topics. Today we are going to talk about the biofortification for human health. I remember one thing with this topic. In some of the movies which we were seeing in our childhood days, we have seen that some of the tablet is being given to hero, and hero has been energetic without taking food for long times, like Star Trek kind of the movies you people might have been remembering. And probably biofortification seems to be a one step in that direction only. Maybe in the future we may not be requiring to eat food, and probably the biofortification will be working to solve our all energy and nutrition problem. I think Dr. Debashish Bakshi will throw light on this topic more. Another topic which was supposed to be spoken by Dr. Saibal Chakrabarti, who is not being with us for some reason, and I really appreciate Dr. Shipra Bhardwaj, who is a very versatile writer as well as a speaker, and she has been agreed in a very small span of the time to speak on this topic, and she has prepared her topic on herself, and now she will be speaking on xenoestrogens in food and how does it work to lead adrenal failure. So this will be the topic of Dr. Shipra Bhardwaj. And third is genetically modified food, risk and benefit, which will be talked by Dr. Arun Kedia, who is the prominent metabolic physician and speaker from the Chhattisgarh. Am I right, Dr. Arun?
Yes, sir.
Yeah, so he is a very prominent physician from the Chhattisgarh, and I am really very happy that he could be able to join our American College of Physicians forum today. And definitely we shall be benefited of his knowledge, and he shall add value to our existing knowledge. With these words, I welcome you all and hand over the podium to Professor Narsingh Verma. He will guide the symposia in the right direction with the help of Professor R.B. Singh. Over to Professor Narsingh Verma.
Thank you, Dr. Anuj Maheshwari, for making this concept the whole month, the program on nutrition, because this month is important for nutrition. In September, the whole month is dedicated for the nutrition, the most important aspect of the human life. Not only human life, whole animal kingdom, you can say. So during this month, we have series of programs. And today's program is very, very innovative. And as the name suggests, innovative concepts in nutrition. Dr. Maheshwari has already talked about that, what topics were allotted to the different speakers and how they are going to talk about this. And I am very sure that these topics will be very interesting and very useful, because all the three topics have their own significance. Biofortification, as Dr. Anuj has already talked about, is also the past and future, I will say. In past, because in our own culture and religion, we have enormous stories that the persons were living with just very few type of nutritions, maybe because of that, which is coming again in the form of the biofortification. And similarly, this Xenoestrogen is very, very important, because most of the time, a lot of things are going to the earth and coming back to us in the form of grains. So this problem we are facing, and that is affect our whole hormonal system. Today, Dr. Shipra will be talking about the adrenal failure, and Dr. Arun Kedia will definitely have a different type of topic. And I'm very sure that we all will be enriching by this. So not further delaying, I invite our moderator, Professor R.B. Singh, sir, to come forward and ask one by one our speakers to deliver. And Dr. R.B. Singh, sir, with opening.
Thank you very much, Dr. Verma. Good evening, everybody. I'm grateful to Dr. Governor, Dr. Anuj Maheshwari, for giving me an opportunity to moderate this session. Ladies and gentlemen, as you know, that United Nations Organization has proposed Sustainable Development Goals, which cannot be served without the knowledge of nutrition, because 49% of the deaths occur because of the lack or deficiency of nutrients or energy in the diet. And this session is devoted to some part of this, because genetically modified foods and also fortification of foods are both important in providing nutrients to the diet. And now we go to the first speaker. Can you show me the slides, first speaker,
Oh, sure.
so that I can read your introduction?
Am I sharing it all right?
Yeah, but I'm not seeing your bio data in this.
Oh,
It
It is written
biodata.
there, sir. It is written there, sir.
Oh, but that slide is not shown to me. So perhaps Dr. Verma, can you read those bio data?
So, sir, the first topic is biofortification of foods for human health. And this topic will be delivered by Dr. Devashish Bagachi. Dr. Devashish Bagachi is PhD and has so many other degrees as well. And he is College of Pharmacy and Health Sciences, Texas Southern University, Houston. And he is very keen in attending so many programs hosted by us and Department of Botany he has worked, and also he has some original research to his credit in the area of nutrition, and is very fond of publishing various books on the nutraceuticals, and I have been the author along with Dr. Maheshwari in his books, and I've seen that how versatile his knowledge is. So I think he will speak when he will talk about biofortification, then you can realize that what knowledge he has. Thank you. And I will invite Dr. Devashish Bagchi for his deliberations.
Thank you.
Thank you. Thank you so very much, Dr. Maheshwari, Dr. Bharma, and Dr. Singh. Now, we are going to talk on biofortification of foods for human health. Now, why it is frozen? Okay.
Yeah.
Okay.
Okay.
Fine.
Now, first of all, I would like to give a disclaimer that our nutrition products or nutritional supplements, we always give a disclaimer that these statements have not been evaluated by the FDA. These products are not intended to diagnose, treat, cure, or prevent any disease. These supplements provide nutrition-based benefits that promote the structural and functional competence of cells, organs, tissues, and systems in our body. Now, when we start long ago, Hippocrates indicated, let food be thy medicine and medicine be thy food. And we are putting his flags on and running forward. And so long, we have developed several nutraceuticals, supplements, as well as functional foods, which demonstrated wonderful benefits to human health. Now, whenever we talk about why we need these supplements, now, diverse bioenvironmental stressors, they are hovering on us all the time. What are those? Gasoline exhaust, and gasoline, its use and the exhaust, now it is increasing day by day. Environmental pollutants, pesticides, fungicide, insecticide, polychlorinated biphenyl, diverse heavy metals, then industrial chemical waste, endotoxin, and diverse biological factors. Sedentary lifestyle, they also have an impact on obesity and human health. Alcohol, caffeine, and tobacco smoke. Now, the other indicate factors include advancing age. We are all getting young, right? Okay. Now, physical and emotional stress, those are increasing. Consequences, we get damage on our lipids, lipid injury, lipid peroxidation, protein damage, DNA damage, and damage on the macromolecules, and changes in the membrane microviscosity. Now, I am showing here a picture of neurodegenerative disorder, which is increasing alarmingly. And we edited a book on metal toxicology handbook, where you can see that different effect of different toxicants. And Dr. Bharma, he also contributed a very eminent chapter in this book. We also edited a book, Antioxidant and Functional Foods in Neurodegenerative Disorder, how we can use it in prevention. Now, good nutrition is essential to maintain aerobic cellular respiration, homeostatic regulation of adaptation, rehydration, refueling, repair, and protection from structurally diverse infections and cellular injury. It maintains the best possible nutrition and metabolically balanced state, achieve increased immune competence, and boost our host defense. Now, healthy aerobic cellular metabolism effectively improves morphologically, hematologically, and rheological properties of the blood and ultimately boost immune competence and overall health. Most importantly, improve the brain integrity and functions. Now, here we show a number of supplements: vitamin C, vitamin E, carotenoids, minerals, zinc, manganese, copper, and selenium, and polyphenols, flavonoids, including phenolic acid, stilbenes, that is the wonder molecule resveratrol, and lignans. And they have shown that they modulate our endothelial function, boost nitric oxide, and reduce antioxidant status. And now we are talking about a number of wonderful Indian spices. One is cinnamon. Cinnamon, it prevents the entry of the virus. It has been published by an Australian scientist in 2020. We also know the effect of ginger root and the solid Indian gold. We mentioned turmeric or curcumin as the solid Indian gold. And you remember, our grandma used to give it with honey. Why? To enhance bioavailability. And now we are formulating curcumin with bioperine. Bioperine, it comes from black pepper seeds. And this product is being in USA circulating wonderfully. We also know the benefits of pine cone extract, the aloe vera extract, the quercetin, the flavonoid, and bromelain coming out of the pineapple stem, and L-lysine. They have significant benefits and impact on human health. Vitamin C, as mentioned by Linus Pauling, that this is one of the wonderful antioxidant which can give us protection, and the dosage is now being used 1,000 milligram. We also have 3 wonderful neuroprotectant, which we forgot, but India, the invention came about 3,000 to 4,000 years ago using Jyotishmati, which can boost our brain power. Second, next is Shankha Pushpi, and in Bengal we call it Aparajita, and which is undefeated. And third is Bacopa. We developed a novel supplement by combining the Shankapushpi, Jyotishmati, and Huperzia serrata. Last year, we published a paper. And rosemary, a unique source of a triterpenoid, ursolic acid, and it has been sold as a wonderful supplement for human health. There are several combinations has been developed by the Indian nutraceutical companies Palmostrong, a combination of quercetin, ursolic acid, and vitamin C, which demonstrated protection against viral infection. Now, we all know about zinc supplements. Once zinc was banned in India and other parts of the world, but zinc supplements, it boosts, if it is used in the right amount, it boosts the antioxidant and immune competence. It is a wonderful membrane stabilizer, protects against viral infection, activity of a host of zinc metalloenzymes, skin and connective tissue metabolism and repair, wound healing, then prostate health, then ocular health, synthesis of DNA, RNA, collagen, antioxidant enzymes, you name it, SOD, catalase, glutathione peroxidase, protein, fortification and replication of cells, and gene expression. We publish, and it boosts the sense of taste, night vision, and healthy epithelial cell. We published a number of papers starting in 1996 all the way till 2008. And now we talk about vitamin D. Now we get it free of cost if we expose ourselves to vitamin D. But we don't have the luxury of time. So vitamin D deficiency has been demonstrated, boost the inflammatory responses, boost immune dysfunction, predisposing severe infections, and increase the vulnerability to infection with enveloped viruses, including retrovirus, herpes virus, hepatitis, dengue, and even our favorite COVID-19. Now, global data correlates that severe vitamin D deficiency with COVID-19 is associated with coagulopathy, reduced platelet count, prolonged thrombin time, and disrupted immune response and increased mortality. So these are all very alarming. We published a paper where we showed that vitamin D is instrumental in reducing cytokine storm, and it boosts the immune competence, it boosts antioxidant, antimicrobial, and anti-inflammatory efficacy by modulating innate and adaptive immunity, enhance the expression of genes associated, and maintain cell junction integrity. So these things we published in a publication in 2020 in Journal of American College of Nutrition, and we are coming out with a book, Nutrition and Functional Foods in Boosting Digestion, Metabolism, and Immune Health from Academic Press. It will come out around end of November. I did it with Dr. Sunny Ohia, the provost of Texas Southern University. And probiotics and psychobiotics, it can help in the management of human health. Especially, it is very good in attenuating dysbiosis. What is dysbiosis? As we all know, altered gut microflora predisposes abnormal inflammatory status and increase the susceptibility to disease. Probiotics maintain symbiosis in the gastrointestinal tract and boost immune competence. And symbiotic state helps the control the severity of disease via the gut-lung or gut-brain axis. And here you can see symbiosis, dysbiosis, and there is a balance. And probiotics and psychobiotics can help. We published a paper in Archives of Medical Research, which just came out in Archives of Medical Research. It is no longer in press, it is now out. Probiotics and psychobiotics, that has been shown, it is instrumental in boosting memory, it boosting the cognition, it attenuates or ameliorates behavior, and it reduces anxiety disorder. And it can also help against stress, and it can help in boosting the immune system and reducing the gut pathogens. And incidentally, there are more neurotransmitters in our gastrointestinal tract than in our brain. This is very, very interesting. You can see the literature. And we publish this in Archives of Medical Research. I will be happy to share you a copy of this paper, and we hypothesized a synergistic efficacy of probiotics and vitamin D. I am going to discuss the salient features. Probiotics secreting lactic acid will reduce the intestinal pH and thereby increase the vitamin D absorption. Selected probiotics increase the VDR expression and its transcriptional activity. Increase the expression of cathelicidin, which is an antimicrobial peptide, and increases Paneth cells and expression of defensins to protect against viral infections. Now, we are going to talk on a epidemic, obesity. It is a worldwide epidemic. Can we use nutraceutical supplement and is there any efficacy? We are going to discuss that in a minute, but you can see the worldwide scenario on obesity and extreme obesity, and it is going up alarmingly. No, effective body recomposition and misconception of traditional weight loss strategies. Now, the term weight loss, it is an unreliable metric. Now, we, after research for several years, we came up with a concept. The primary focus of heaviness of the body does not provide an accurate perspective for evaluating healthy changes in metabolism, body composition, or size. Fat is the lightest. Fat is the lightest and usually the last to go in the body decomposition process. Thus, short-term expectation in weight loss are erroneous. Our body is genetically pre-programmed to survive and has 3 forms of survival insurance. What are those? Fat, sugar as glycogen, and 3, the water. When the body is put into a survival mode, that is survival panic, upregulates survival insurance storage of these components and retards energy expenditure and increase energy conservation. Here you can see all the intricate factors associated with obesity: environmental toxin, genetic predisposition, immune system distress, aging, stress, enhanced production of reactive oxygen species, impaired aerobic metabolism, anaerobic metabolism, boosted immune response and immune function, dysbiosis of gut microflora, that is all being affected by sedentary lifestyle or poor dietary habits. And all this leads to the central point, obesity, and it is increasing day by day. Now, reversing adversarial gene expression of neurotransmitters and hormones involved in fat metabolism. This is possible, and there are several intricate factors. One is leptin. Leptin deficiency or resistance can cause a number of diseases: infertility, obesity, T1D, T2D, other insulin-resistant disorder, and the Rapson-Mendenhall syndrome. Ghrelin, the other hunger hormone, is survival panic-induced hormone that increase appetite, promotes cell longevity and tissue protection. And reproductive hormones, adipose tissue-derived estrogen, exert anti-inflammatory activities in the periphery and the CNS to protect from inflammation. Hypothalamus regulates energy homeostasis and ventrolateral region of the ventromedial nucleus. Arcuate fibers respond to hormones and other signals to regulate the energy homeostasis via energy intake and expenditure. Thyroid hormones, T3 and T4, play vital role in body weight regulation and energy homeostasis. Neurotransmitters, serotonin and dopamine, and reversing the adverse effects of premature aging on fat metabolism, and reversing the adverse effects of AMPK deficiency on fat metabolism. To further state that poor dietary habit, inadequate physical activity or sedentary lifestyle, overindulgence of food, high-fat diet, sugar, and salty foods, they all lead to obesity. Other important factor, exposure to environmental toxin, agribusiness practices, livestock treatment. These are going up day by day. Processed food, food additives, flavor enhancer, preservatives, and colorants. They are contributing immensely. And what all it does, digestive malfunction, it dysbiosis, and boost the survival panic, impair gut-brain interconnectivity, and impair cognition, boost taste tolerance, sadness, increase energy conservation and fat storage, and boost the anaerobic metabolism, inflammatory response, fat tissues that act as an organ unto itself, sending our chemical messenger that inhibit healthy fat metabolism and increase fat storage, retarding protein conversion into muscle. This is very alarming. Increase oxidative stress and oxidative tissue damage, boost immune distress and exhaustion, trigger survival panic, premature aging, impaired aerobic metabolism, increase anaerobic glycolysis and metabolism, and reduce AMPK. As we know, AMPK is abundant in every cell in the body, which triggers burning of stored fat for energy and induces increased fat storage. We developed a novel protozoan TARCAP21 phytoceutical intervention. We are going to discuss that in a minute. These are two of my books on obesity, about a 1100-page book, and another book we did it with childhood obesity for academic press. Now, what is so great in TARCAP21? We protozomed it. We developed a technology, proteasome technology, and we patented, it is patent pending currently, to boost the bioavailability and absorption. And we developed the formulation of TARCAP21 using D-ribose nicotinamide complex, which improves NAD and ATP availability, optimizes cellular energy production, gynostemma pentaphyllum, activates AMPK, reduces pro-inflammatory cytokines, and nullifies blood acidity. And we used camu, Myriscaria dubia, rich sources of vitamin C and betulinic acid, reduced pro-inflammatory cytokines. We used alpha-glyceryl phosphorylcholine, enhances acetylcholine release, and increases hepatic fat oxidation, which is a key step in regulating obesity. Rich in antioxidant mediates activation of TGRF nuclear receptor involved in the prevention of diabetes, and we used Camellia sinensis, a purple tea, a rich source of different group of polyphenols, and it is a strong antioxidant. We developed the formulation which boosts the different type of antioxidant potential, restored physical homeostasis and aerobic metabolism, and increased oxidative damage protection that helped in reducing the obesity. I am going to discuss that in a minute. We developed a concept validation study of TARCAP21, commercially known as Stimrox. It was supervised by Dr. Morrison in 9 subjects, male 3 and female 6. And we used age group 37-70 because this is the age group they are more prone to obesity. And we did a 21-day study. 8 subjects took 1 dose in the morning in an empty stomach, 6.75 milligram, 21 days. And the 9th subject took 2 doses of 6.75 gram per dose. First dose in the morning, second dose in the evening in an empty stomach over a period of 21 days. All subjects provided and maintained daily diary, and they were specifically instructed not to change their lifestyle. And we measured the adverse events. And here you can see on the multiple anthropometric measurements, we believe that body weight is important, but measuring the chest, measuring the upper arm or bicep, hips, thighs, waist, and belly, these are equally important in reducing their size. which is important in reducing the integrate parameters of obesity, and we do not want it fast. We want it going slow and steady. And here you can see in all these, there is a significant loss in body weight, but more so in the chest, in the upper arm, in the hips, thighs, and waist and belly, there was significant reduction. And we measure the energy level, we check the mood, we measure the satiety, we did the sugar craving. Here you can see all the subjects, and these are the doses they used. We used the energy level increased in almost more or less in all participants. Mood was improved, satiety level was improved, and sugar craving was reduced, at least in 7 of them, however, no effect in 2 people. And one subject, they said he didn't report anything. Sleep was improved in almost all the participants. Overall health improved, and no adverse event was observed. Incidentally, we used this TARCAP21 in a type 1 diabetics, and we found that it is working interestingly. And in 2 months, she lost about 11 kg body weight. It is very interesting. We are getting more data and more information so that we can present it safely and regulatory, make it friendly. Now, overall, this 21-day concept validation pilot study exhibits TARCAP21 enhanced healthy body recomposition observed as all subjects experienced reduction in size of various body parts, increased energy level, elevated mood, enhanced satiety, reduced sugar craving, and improved restful sleep and improved overall health. They got more energy so that they can work more. And overall diverse functional food exert potential health benefits, and they get enhanced healthy gut microbiome. These are the 2 books we published. One, we did nutraceutical and functional foods in human health and disease prevention, came out in 2018. And we are coming out with a book, Microbiome, Immunity, Digestive Health and Nutrition. I am doing it with Bernard Downs, my colleague and my beloved friend. And finally, these are my contact details. If you have any question, I will leave my slides with Dr. Maheshwari and Dr. Bharma and Dr. Singh. You can always ask me question. These are other 2 books I edited. One is Immunity and Inflammation in Human Health and Diseases. And the other book I did it on chronic inflammation. One from Elsevier Academic Press, one from CRC. These can explain all these immune competence, how this can be enhanced by reducing this inflammatory response. I thank you very much, and it will be my pleasure to address any questions you may have. Thank you.
Thank you very much, Dr. Bagchi. You have proven beyond doubt that you are very, very learned in the field of nutrition. So can you tell us
Sir, questions we can have in the last when all the 3 speakers have done it. So that will be, but you can ask, sir.
One question, because I have to go. So may I ask you about the role of some nutrients in developing the yellow gouty mouse? There is a deficiency of some nutrients like pyridoxine, B12, and the product during pregnancy, and the products come out that is yellow gouty mouse, which has a yellow fur. There is no change in the gene sequence, but there is a alteration in epigenome. Nutrition epigenetic interaction and the production of metabolic syndrome in a yellow agouti mouse experiment.
Yeah. It is very interesting, Dr. Singh, you know, that we worked on the that particular strain of mice, and also we did work on other obese mice. And what we found that they have already a gene, and there are a problem on that particular gene, but that does not mean we cannot rectify it. And more so with personalized nutrition and with personalized supplementation, you can definitely modify that gene at least partly. And we worked on another group of mice where, you know, those are fat mice. And they are so lazy that if you push them, they will move maybe 1 centimeter and sit right there. And they have more fat and less, muscle. And what we found by treating them with chromium supplement, that they develop the muscle cell within the fat tissue. And we are working, my one research group is working how those muscle cells can be developed in the obese mice, and then we can translate it in personalized nutrition to the human body. I will send you, our publication came out in 2009 on that aspect, on the obese mice.
Thank you very much.
Thank you.
Dr. Bakshi. I know you are connected with Dr. Basu in Edmonton, and perhaps you are very, very learned. So now we go to the next speaker. Some more questions would be asked to you, so please be here.
Sure.
Dr. Bakshi, please
Sure.
stay
I will
with
be
us
available.
because we will have discussion in the last. So please stay with us because lots
Sure,
of
sure. I will be.
also ask the question, sir.
Sure.
Okay, sir. Okay, Arvind Singh, sir, please proceed.
Yeah, please, next speaker. Can I have your slides, please? Next speaker.
Sir, I will introduce her.
Yeah.
Shipra
Shipra, okay, yeah.
is a PhD from KGMU, and she has most of her work in the field of chronomedicine. And today she has agreed to talk about xenoestrogen in food and adrenal failure. She is a woman scientist at present in Department of Health Research, Department of Physiology, King George Medical University, and she is credited with more than 30 papers in the peer-reviewed index journals, and very active scientist. With this brief introduction, sir, you know her since very beginning. Your blessings are with her. Dr. Shipra, please unmute yourself and start your deliberations. Unmute yourself. Yeah.
Thank you, sir.
Yeah.
I think my slides are visible.
Yeah, they are visible. Make it in PowerPoint. Yeah. Good.
Thank you, sir. As sir has given me this topic yesterday only, but I would like to tell you that my initial research just after I'm master's have started from the field of toxicology. So a big part of my initial research was purely on these endocrine disruptive chemicals, and xenoestrogens are basically a part of those chemicals only. So as the FDA suggests, and it has defined the xenoestrogens, they are the specific estrogens. They are the chemical which act like a specific estrogen in the body, and these are sometimes harmful toxin that imitates estrogens and dramatically increase the toxic burden of certain hormones. All the plastics and cosmetics and all the pesticides and all the adulterants in the food industry and colorants, all these chemicals are 90% in the category of endocrine disruptor, and that too xenoestrogens. Basically, xenoestrogen classification, when we go into the detail, they are some of the xenoestrogens are natural that are called as phytoestrogens, mycoestrogens, and some drugs of abuse. Phytoestrogens are therapeutically used in various diseases. So they are very you can say it's a beneficial part of the xenoestrogens. Mycoestrogens are somewhat they are the fungus which grows along with the certain crops, so they are very injurious to health also. And there are certain drugs. So in all the category of xenoestrogens, only phytoestrogens which are used in medicines, like in breast cancer medicines and in hormonal medicines, especially in cases of endometriosis, etc., they are used. So except phytoestrogens, xenoestrogens are comes into the toxic category of EDCs. So basically, in toxicology, xenoestrogens are always called as the endocrine disrupting chemicals. According to the FDA, it is defined as the exogenous agent that interferes with the production, release, and transport, metabolism, binding action, or elimination of natural hormones, and which are responsible for the maintenance of homeostasis and the regulation of the developmental processes. Few examples are all the pesticides which are used in agriculture. There are certain drugs like diethyl, bestol DS. There are certain chemicals like polychlorinated biphenyl, PCBs, and all the plastic material which contains bisphenol A. There are heavy metal cosmetics, textile, paints, and lubricants. How they reach in our food? The most important question of this talk. First, it's initially started from the agriculture, from the first phase, right from when the farmers start sowing the seeds. It started mixing the soil with lots of fertilizers, and most of which are chemicals nowadays. So the base start from there only, and then in all the phases, right from the sowing, growing, spraying from the fruits, then until it comes into the plate of the consumer, it is filled with the lots of ADCs and xenoestrogens. 95% of xenoestrogens, or we call EDCs, are because of the agriculture practices and mostly how they store them. That is the main point. 95% of it are because of the agriculture techniques, and rest 5% nowadays the cookwares we use, it contains Teflon, and if it is of not food grade quality, or it is sometimes it is rough, or somewhat some people use plastic containers which contains bisphenol A for the food storages. So 5% xenoestrogens comes from here. Now, if we talk about, I've taken one example while I was searching that the most important food which we call is that with apple occurred. I found it this example. Actually, it has been said that an apple a day keeps a doctor away. But when I just studied the whole apple orchid calendar, that in autumn they spray herbicides, in winter they spray fungicides and insecticides, in spring fungicides, and then summer again insecticide. So whole apple orchid calendar of whole season includes lots of pesticides. So it is definitely just spoiling this definition. Other ways of xenoestrogen exposure are definitely water, because all the sprays and all the pesticide spraying, herbicide spraying, all the chemical spraying ultimately goes into the river water or are absorbed under the soil. Then comes food. As I said that all the fruit, vegetables, grains, all contain some amount of pesticide residue even after they have been washed or cooked through roughly. Then bioaccumulation in animal products like milk, fish, meat, and all the dairy products. Now, how we have calculated these, that these are coming into our food. Then this comes according to the what by the pesticide, EDCs, and chemical residue estimation. As defined by the World Health Organization, simply I would tell that pesticide residue refers to the pesticides that will remain on in our food after they are applied to the food crops, or all the chemical or EDCs that are being uh, being being given into that food. All the regulatory bodies of all the countries have allowed the maximum allowable levels of these residue in the food. In India also, there are maximum residue limits of each and every chemical which is used in agriculture and food. There is an acceptable daily intake ADI, and the level of food additive uses varies by country because all the agriculture situation of different regions are different. So pesticide residue ultimately you can found in, I'm saying again and again pesticide because actually these EDCs are pharmacologically and toxically comes into the category of pesticides only, and then it is diverted into herbicides, weedicides, or food adulterants or different colorants. So each and every food, especially in grain, cereals, fruit and vegetables, you can find high amount of pesticides and EDC residues. This is the Times of India photograph, which I took around 2 years before for a presentation. In this also, you can see that if we compare the maximum residue limit of some pesticide, which are actually banned all over the world. So in India, it is the comparison of the some pesticide used in India and European Union. See, malathion in cauliflower, it is 3,000 more than the maximum residue limit. DDT, which is almost banned, we can find in tomato, 3,500 maximum residue limit. Then parathion methyl, the most importantly used vegetable in all the diet, potato, 1,000 MSL. Endosulfan, it is a very potent carcinogen ADC. We can find it spinach. And this is the 2 years later data. EDC now you can find in every food, even in soft drink, in bottled drinking water, and even in breast milk. So this is the pesticide residue occurs in different food: oats, wheat, cereal, rye, bread, rice cakes, any food you name, and the pesticide residue is here. This is an important chart which I found in the FSSI website. The maximum residue limit of the pesticide in different food. You can see that in nearby Allahabad, it is recently found that tomato, the DDT, which was restricted use, and it is a carcinogen endocrine disruptor, and it have xenoestrogenic effects. It was found in severely 0.01%, but it was found, and this is restricted in India. Like here in nearby Allahabad, Gorakhpur, Faizabad, you can find heptachlor. All these chemicals are actually banned in India, and still the pesticide residue is found. In breast milk, since EDC can cross the placenta, they have been detected in the amniotic fluid, and of course in breast milk also. And organochlorine pesticide, which are the potent EDCs, have been detected in breast milk, which including DDT and heptachlorbisphenol, and there are HCS isomers in the contaminated areas, especially in UP and nearby areas. So potential health hazard of xenoestrogens are definitely: first is the acute toxicity, which includes like respiratory tract irritation, digestion problems, allergy, eyes, skin irritation, nausea, vomiting, and there are chronic toxicity effect which include Parkinson disease, all type of cancer, including lymphoma, and there are some asthma, depression, anxiety, and ADHD also. As far as endocrine disruption is concerned, these chemical causes estrogenic effect, anti-androgenic effect, anti-thyroid effect, and anti-progestin effect, and there are lots of teratogenic effect There are also embryotoxic, as I told, and teratogenic effect also, and they can disturb the development of embryo or fetus. My senior did a research during that time on the pesticide I just showed in the last slide, chlorpyrifos. She studied in the 3 generation studies. She studied the residue of chlorpyrifos. It was a teratogenic study in the mother and fetuses. She found it in all the 3 generation study. So you can just think that what the effect of these chemicals are. And of course, they are the carcinogenic. Almost 300 pesticides are potent carcinogenic at certain doses. How these xenoestrogen biotransformate inside the body? First, they involve a combination of several chemical reactions, including oxidation, reduction, hydrolysis, and then it causes oxidative stress and the free radicals increases in the body, and ultimately the cancer prognosis and other things carry on. Biotransformation of some organophosphate pesticides involve oxidative activation, and they reaches many organs and tissues, and all these accumulate into the adipose tissue. They are eliminated through urinary, biliary, and fecal material, and they are also found and excreted in milk, and it has been experimentally proved because the doses of DDT dialdrin have been found in mother breast milk. The mode of action of EDCs are definitely because as they couple with the hormone receptors, these endocrine receptor couple with the hormone receptor, and thus mimic or block the transcriptional activation elicited by the naturally occurring circulating steroid hormone by binding to steroid hormone action. Now, this is the mode of action for fungus versus human. I have taken the example of fungus because mostly for agriculture purpose, these xenoestrogens are used to inhibit the effect of fungus for the crops actually. So as far as fungus is concerned, it affect on fungus by inhibiting this ergosterol synthesis, and thus the progression of fungus inhibited. So the crop is saved by the fungus. But in case of human, because the traces of it ultimately goes into the food and then finally into the human gut. And what happens there? Then all it combines with the hormone receptors, and this is in the and interfere with this process. Like this is the same process in both the entity. The first step 14 dimethylase, then the cholesterol is made, progesterone, and here it inhibits the synthesis of aromatase enzyme, and ultimately it disturbs all the later pathways. Now, why in this talk, why we have taken adrenal gland? Because it is more vulnerable to toxicant because of the following reason. First, it is highly vascular gland with high rate of blood flow. Second, mechanism for uptake of storages of lipoprotein and ultimately associated lipophilic toxins also. Then free radical generation during steroid biosynthesis is potential for bioactivation of toxicant by cytochrome P40 enzymes and other enzymes are there. High membrane content of unsaturated fatty acids. Substrate for lipid peroxidation is there, and multiple site at which functions may be influenced, whether it's in cortex area or the medullary area of the adrenal gland. Xenoestrogen ultimately attacks the adrenal gland via the modulation or inhibition of enzymes involved in the steroidogenesis by inhibiting aromatase, as I told in the previous slides. Also, along with aromatase, 5-alpha reductase as well as 3-beta, 11-beta, 17-beta hydroxysteroid dehydrogenases are also inhibited. It also inhibits steroid regulatory protein star there, and ultimately altering different steps of steroidogenesis in adrenal gland. So ultimately, this is the normal steps of the hypothalamus and adrenal cortex pathway. This is the normal one. Ultimately, when oxidative stress because of any kind occurs, this pathway is disturbed by the increased glucocorticoid secretion and adrenal cortex hypertrophy, which result in the stress secretion of glucocorticoids. And in more severe condition, when the toxic inhibition occurs, then compensatory ACTase secretion is done, that ultimately either it abolish or decrease the glucocorticoids. And ultimately, the result is the same, adrenal cortex hypertrophy, which again results in adrenal toxicity and impaired stereogenesis and ultimately adrenal failures. This is my own research study on imidacloprid. Imidacloprid chemical is actually an ADC. It is also a xenoestrogen, and it is a quite fewer big study of my initial research career. In this study, we did that on 90 days over toxicity of imidacloprid in female rats to find out the effect of biochemical and histopathological and morphological evaluation. This was published in FCT and it was my first paper. This study result almost so that in 90 days, we gave the 4 doses of imidacloprid, which was an ADC, of 5, 10, 20 milligram per kg per day body weight in female rats. In the results, we found that the body weight gain what observed at 20 milligram kg per day, and at necropsy, the relative body weights of liver, kidney, adrenal was significantly increased. In this study also, I found that adrenal gland and kidney weight was increased at 20 milligram kg per day, which was the highest dose. And also all the toxic symptoms was seen at the higher dose, not in the 5 and 10 milligram kg per day dose. So ultimately in this paper, we concluded that for this EDC or this xenoestrogen, which was the imidacloprid, it is sprayed in India for these crops like rice, cereal, millets. Nowadays also it is not banned. It is quite, I'm talking about milligram, 20 milligram kg per day. In agricultural use, it is spread in kgs, not in milligrams. And 20 milligram per kg per day body weight. This is the clinical chemistry parameters. We found that there was the significant elevation of serum GOT, GPT, means liver enzymes, and decreased activity of acetylcholine in serum and green. And the spontaneous locomotor activity in roads were decreased at highest dose exposure. So ultimately, as far as adrenal toxicity is concerned, so this xenoestrogen also increased the relative weight of kidney and adrenal. So in this study, we suggested that 10 mg per kg per day dose may be considered as no observed effect level dose for female rat. So we can calculate the dose for human and for the frog. So during 2011-12, we gave this concept that 10 mg per kg per day should be used in imidacloprid, and we have sent that to, because the study was done in Industrial Toxicology Research Center, which is a government institute. All the result was sent to government, and the doses were decided, but still imidacloprid has been used in kgs in agriculture in India. So ultimately, when we talk about the conclusion, why these ADCs, adulterants, are used. First, mostly the dose concept is not clear. Not only for uneducated or less educated farmers, not even the workers, the administrator, the dose concept is not clear. And in India, farmers are not much educated, of course, and mostly they rely on what pesticide industry, dealers, distributors, shopkeepers tell them, and they mostly go for cheap banned pesticide rather than EPA, FDA, or any government agency approved pesticides. Reason for this, because the Indian pesticide market, that is the chemical industry market, is the fourth in the world. In the slides. prevention, because ultimately we as a health workers, we should just go for the prevention, how to prevent it and how to just save from this. First, we have to go for the prevention at the local level, use pesticide only when benefits outweigh the risk of pesticide and necessary. Then there should be community level prevention like integrated pest management, community activities, and educating the farmers. Then there should be prevention at national level by education campaigns and all. Then international level. Internationally, there are many organizations which work on it, like, and there are many more. And also in United Nations and other 50 countries have banned many like alveolar DDT. These are the potent endocrine disruptor, but still we don't know from where they are being used in it. Now prevention. These are the these prevention methods are in other hand. What is there in our hand? That we can wash our food and wash it right, not using the chemical which are being produced, which are being given in the market. We can wash them with 2% of salt water and most contact pesticides. And almost many pesticides can be easily removed by only washing them from 2% salt water or sodium carbonate solution. Then avoid long-term storages of cereals and grain. In India, the most important thing is that people store grains for even 1 year, 2 years. Means ration. The process of collective ration in India. What happens? That the crop which is being sprayed from the root level, from the soil level, then ultimately when it comes into our home, it again goes to the PTCs and chemical exposure, because to store it for the 1 year, you have to spray or you have to spread some pesticide or insecticide or fungicide or something like that. So ultimately, they are also diseases. Ultimately, they are also Xenoestrogens. Then, if by cooking soak cereal and pulses in warm water for overnight, before cooking, this will eliminate hard water soluble EDCs or pesticides up to 75%. And for rice, instead of cooking in pressure cooker, use boiling method as we done before in the ancient time in India. Go organic, but the problem in going organic is that they are most expensive. Because the problem in India is that if you want unpolished dal, then you have to pay more than the polished dal, and that is not it. Then there are role of our scientists and health professional implementation of this toxicity. First, diagnose interest, then we have to do intensive research, and ultimately we have to publish and educate them. And of course, prevention is better than cure. Now, I think this is the biggest estrogens and all the EDCs, not only them, but all the EDCs and pesticides, and what we are eating in food. Calorie, nutrition, I think it's more beneficial than we eat fresh food without pesticide contamination or any chemical.
Thank you. Thank you, Dr. Shipra Bharadwaj. Perhaps you have taught us everything, but still, if you can answer any clinical study on the effect of pesticides in pregnant women on infants or fetus. Are you aware of any study? It is okay, don't worry.
I can't hear your question clearly. Actually,
effect of EDC or pesticides in pregnant women on fetus and newborn. Anyway, you read it and tell us next time. Now we go to the next speaker. Next speaker is a very, very renowned Dr. Kedia, and he would be talking about genetically modified foods. His experience is very well experienced, more than 10 years, and life member of the association, various association, president of API in Raipur, district coordinator of the RSSDI, that is a diabetes organization, and part of various clinical trials he has taken, and publications in journal, and also book chapters of national importance. May I request Dr. Kedia to go ahead with your subject, genetically modified foods.
So good evening, sir, and thank you for your kind introduction, sir. First of all, I would like to thank Dr. Anuj sir and Dr. Verma sir to give this opportunity to speak on this nutrition platform, because as clinicians, we hardly ever take care of nutrition of the patients. At the same time, I'm very honored to be sharing the platform with Dr. Singh sir, Dr. Bagchi sir, and Dr. Shilpa madam. And I would be happy to express my views on the genetically modified nutrition. So the theme for today's discussion was innovative concepts on nutrition. And we know that without innovation, we cannot advance. And that is what is genetically modified nutrition. It is one of the most promising innovation in the field of nutrition. And probably, as Dr. Bagchi sir said, that we want a sustainable development in nutrition as per the WHO. And probably genetically modified nutrition would be one of our major steps in providing a sustainable nutrition. Now, however, whenever we have a new innovation, we have certain risks as well as benefits. So in next few minutes, I would be speaking about the benefits and the risks of genetically modified nutrition. So what is genetic modification? So it is a special gene technology which alters the genetic machinery of living organisms, be it animals, plants, or any microorganisms. So you do a combination of genes from different organisms in the form of recombinant DNA technology, and that resulting organism is called as genetically modified or genetically engineered or transgenic. So technologies for this genetically modifying foods, they have offered dramatic promising for the meeting some new areas of greatest challenge for the 21st century. And as I said, that sustainable development for nutrition is one of the targets. So like all new technologies, they would also have some risks which are both known as well as unknown. And at present, all we can say that probably they can be one of the most cost-effective, sustainable, and realistic direction in the field of sustainable development of nutrition. So when we came out with genetically modification of nutrition, the major aims were to nutritionally enhance so as to improve the content and bioavailability of essential nutrients. And in the process, what we are targeting is the hidden hunger. So basically, we are providing the micronutrients and minerals which are essential for the overall growth of the patients. At the same time, we want certain ingredients to be inserted so as to fend off chronic diseases like heart diseases and cancer, maybe in the form of incorporating some polyunsaturated fatty acids and essential amino acids. So going to the history of genetic modification, we know that this technology was first discovered in 1946 in the form that we can do some gene transfer through DNA. And the first genetically modified GM plant was produced in 1983, which was an antibiotic-resistant tobacco plant. And China was the first country to commercialize it. Then we had in 1994 a transgenic tomato, which was approved by the FDA for marketing in the US. And since then, we have been growing further. And today, as of now, we have a total of 35 approvals internationally to commercially grow 8 transgenic crops and 1 flower crop. Now coming to the advantages. So the most important advantage that we see medically is the nutritional enhancement. So let me quote certain examples. So the role model for nutritional enhancement is the biofortified rice, or what we popularly known as the golden rice. Now this innovation came out to provide vitamin A in places where there is inherent vitamin A deficiency, which is one of the commonest cause of night blindness in the underprivileged society. So this golden rice actually appeared yellow because there was a gene incorporated into it, which had carotenoid biosynthesis pathway. And this gene, when was incorporated in the genome, the endosperm of the rice had a good amount of beta carotenoids, which could be transformed into vitamin A into the body and thus could provide a good supplementation. On the other hand, there were development of other rice which could enhance iron and folate supplementation. And there were many other varieties of rice where we could have, where they found that they could enhance the product availability of iron, folic acid, as well as vitamin A through the process of gene stacking, that is incorporating multiple genetic pathways into the same plant. Then we have the transgenic corn with phytase. So these phytase, they are basically enzymes which reduce the availability of zinc bioavailability. So once we have phytase, recombinant microbial phytase, they are used to reduce the level of phytic acid in the grains, and thereby they improve the zinc bioavailability. And as Dr. Bakshi said already, how important is zinc in our diet. Then we have examples of maize, where a triple vitamin fortified maize was produced, which express high amounts of beta carotene, ascorbate, as well as folate, which was developed in the endosperm through metabolic engineering. On the other hand, the corn oil, which serves as a source of lecithin, was also developed. In underprivileged countries, especially Africa, cassava was developed with high levels of beta carotene to provide vitamin A. And then there was another variety which was produced by reducing the levels of toxin cyanogen in the roots, which could improve the iron root uptake and protein accumulation in cassava so as to provide a biofortified cassava. Similarly, wheat was undergone the genetic modification, and celiac disease causing gliadins were lowered from the wheat in the form of genome editing. So this was another technology that was developed whereby the natural ingredients of certain plant-derived cereals was reduced so as to prevent certain diseases. At the same time, level of free lysine, which is an essential amino acid, which was normally scarce in wheat, was developed. Then chickpea, which was capable of accumulating high concentrations of selenium, was developed as an antioxidant source. So these are the certain examples of nutritional enhancement which could be achieved through biotechnology and genetic engineering. Another area is the biopharmaceuticals. So in this, the vaccines and other therapeutic proteins, including monoclonal antibodies, can be produced in the plants. So this is another very important and promising area of research. And today, a variety of therapeutic proteins produced in plants is considerable. And it could range from antibodies against HIV to vaccine proteins against smallpox or other biological warfare threats, and even to an assortment of anti-cancer therapeutic agents for the newly emerging field of personalized medicine. So these are another very important aspects of innovations. And clinical trials are already underway to examine the ability of plant-derived therapeutic proteins to treat challenging diseases like HIV and Ebola virus. So biopharmaceuticals could be another challenging innovation. And if this comes out to be very successful, then we might have vaccines and monoclonal antibodies being delivered through plants. Other advantages could be that these genetically modified crops are highly resistant to diseases, pests, insects, and herbicides. So if we see the example, the insect resistance is achieved by incorporating into the food plant the gene for toxin production from the bacterium Bacillus thuringiens. And one of the commonest production is the Bt cotton, which is insect resistant. The virus resistant was achieved by means of introduction of gene from certain viruses which cause diseases in plants. And the example is ring spot virus resistant papaya, which was grown in Hawaii, and the triple virus resistant zucchini, which is grown in certain areas of US. Then we can have a herbicide tolerance, which is achieved by introduction of genes from bacterium having resistance to certain herbicides. And the examples are herbicide tolerant sugar beet and herbicide resistant canola. So these are certain examples whereby we can improve the production of crops, decrease the wastage, decrease the destruction by natural pests and various herbicides. We also have crops which could have a high tolerance to cold or heat, drought, or salinity, which could be helpful in the context of global warming and climate changes. So all sorts of abiotic stresses could be, one of the ways by which the plants could be genetically modified so as to cope up with the adverse environment. A few other advantages include food security. So we know that the population of the world is growing. There is growing urbanization. So the GM crops, they could provide one of the promising solutions to meet the world's food security needs. At the same time, these GM crops can also provide high quality animal feeds so as to provide a better animal produce, and livestock could be much more healthily taken care of. On the other hand, if we see the economic benefits, then these GM crops, they improve the yield, thereby it could increase the farmer's income. GM crops have a longer shelf life. So one of the examples is the tomato, whereby these tomatoes were found to ripe slowly and they could stay fresh during transportation and storage. So thereby you can avoid wastage. And also GM crops can help reduce the dependence on imports, especially the oil seeds. So these are the few advantages that we could achieve through this biotechnology innovation in terms of having food security. But at the same time, as I said, whenever a new technology comes, we certainly have to take care of the concerns. So one of the major health concerns that genetically modified food could have was introduction of allergens and transfer of antibiotics resistant markers. There have been certain studies which have shown strong correlation between GM crops and birth defects, cancers, kidney injuries, diabetes, autism, and Alzheimer's. But of course, we don't have a direct correlation and a direct cause and effect study so far. So if we see the safety issues of GM foods, there could be safety concerns with regard to genetic modification, safety with regard to new proteins which are produced by these plants. There could be occurrence and implications of unintended effects. There could be gene transfer to the gut microbiota. There could be allergenicity of new proteins, and the influence of food processing is still not known. So coming to the environmental concerns, they could decrease the species diversity. So insect-resistant plants might harm insects that are not their intended target. So there could be destruction of that particular plant, insect species. Then GM technology could result in transfer of genes from one crop to another, creating super weeds, which would be immune to common control methods. So this is another big concern. And the viral genes added to the crops for the purpose of viral resistance could be transferred to other viral pathogens, which can result in new and more virulent virus strains. So all these are concerns that we need to take care while we promote GM crops. We have certain economic concerns because these crops may be time consuming and could be costly process. We still do not have a very high yield of these crops, and these crops would provide a dangerous concern to the farmers with regard to the concern of the domination of world food production by a few companies, because there could be patent rights of these GM seeds. Then the ethical concerns include violation of natural organisms' intrinsic values by mixing among species, and there could be oppositions to mixing animal genes in the plant. So this could hurt certain sentiments of especially the pure vegetarians. So we have certain genetically modified crops in India. Among them, the most important commercially produced is BT cotton, which is insecticide resistant. And this is the only crop that has been granted by the government, and it is the only transgenic crop approved by GEAC, that is Genetic Engineering Appraisal Committee. Of course, after that, BT brinjal and BT asti mustard was developed, but so far the GEAC has not recommended the commercial approval, and the Supreme Court stay has already been put up against all the GM crops in India. So why there are concerns in India? Because we have regulatory concerns. So we do not have a proper assessment of GM crops in India. Most of the GM crops in India were imported, and we had only one food lab in Kolkata under the Ministry of Health, which was not fully equipped. There is a strong conflict of interest because all the safety tests for regulatory approvals in India are conducted by the same party that applies for commercialization of GM crops. And then we have transparency concerns, which are further doubt creating bodies. And there is a tendency to operate in secrecy, which leads to a serious distrust on the government and the promoters of GM crops. So far, GM crops have not taken a very important role in India. So what needs to be done? There needs to be a research in the field of agriculture. There has to be, this has to be achieved only through technology. And this technology needs to ensure that it does not undermine human and environmental health. We need to have a participatory approach of the government with regard to the scientific evidence, and there should be participatory approach for bringing all the stakeholders together to develop regulatory protocols. We should also take care of the farmers' interest, and we must convince all the stakeholders, including the consumers, the environmental activists, and farmers, to ensure that GM technology is one of the best options to improve crop yield and resolve the developing countries' food consuming concerns. So as of now, the status in India says that FSSAI has not allowed GM foods in India so far. And since 2002, only commercial cultivation of GM BT cotton has so far been done. The reasons are that there has been no Indian scientific study carried out so far to study the impact of GM crops on human health. Long-term effects on the human health have not been studied so far. The Department of Human Health has not taken any action with regard to examination of the impact of GM crops on human health. The government should reconsider its decision to commercialize GM crops in the country. And it is very late in the day for the FSSAI to take the decision to label GM foods imported into the country. So all these are very important so as to ensure that the people and the consumers do not have any doubts with regard to GM crops in the country. So we do have done GM testing in India, and we have found that a lot of food samples have tested positive. So as of now, it is not that we are not consuming GM foods, but these are all off-label products. So what are the solutions? The solutions are that there has to be interactions between many disciplines, including the plant breeders, molecular biologists, nutritionists, social scientists, and to a certain extent, even the doctors, to create awareness. And probably this is the reason why September has been chosen as the Nutrition Awareness Month. And I'm sure Dr. Verma sir and Dr. Anuj sir are doing excellent work to create this awareness. We need to develop new crops that are nutritionally enhanced, and they must be evaluated by clinical trials. The select populations who can benefit most from these must be educated so that they understand how these advantages can make a difference in their community health. And there should be cooperative efforts between the government, the industry, non-profit organizations, so as to truly eliminate hunger and to meet a sustainable development in the field of nutrition, especially the rural sections of the country. So to summarize the benefits versus risks of GM modified plants. So on the one hand, we have benefits in terms of nutritional enhancement. We can produce foods that lack known allergens. They can be grown in arid conditions, under drought conditions also. They can produce herbicides to kill the pests. They can improve food supply and agriculture in poorer countries. They can produce crops which have a longer shelf life, and they can reduce economic cost and carbon footprint, which needs less need for land clearing and pesticide usage. Vis-a-vis, we have risk in the form of new traits which could cause adverse health reactions. We need to have removal of those traits. There can be limitation of biodiversity of local environment. There could be cross-pollination which could lead to superweeds. It could restrict farmers from accessing GM seeds, and they may not be labeling of the GM components. So all these regulatory concerns have to be taken care of by the government, and these are the risks that need to be studied for ensuring that the consumers are well educated and they are more confident about the GM crops that they consume. So with that, I end up saying that our knowledge today is limited, but our ignorance is infinite, and we need to keep researching and keep bringing out innovations in the field of nutrition. Thank you, sir, and I would be happy to answer your questions.
Thank you very much, Dr. Kedia. You have very nicely prepared your lecture.
Thank you, sir.
Yeah, genetically modified foods are used in the Western world for the last 2 decades, also possibly in China. So are there any epidemiological studies or experimental studies that have shown adverse effect or beneficial effects of these foods on cardiovascular disease, diabetes, and cancer? Human studies specifically.
Thank you all the speakers and Professor R.B. Singh sir. There are few questions from my side, and other questions will be asked from Dr. R.B. Singh sir. My first question to Dr. Devashish Bagchi. Sir, in biofortification, I have one question which is asked usually in all the conferences. And beaters of this biofortification, they usually ask that by mixing our biofortification, whether the required vitamins, minerals, etc. are reaching to the place where they are required most. For example, if we talk about antioxidants or minerals or vitamins, by taking them, mixing in the food, whether they will reach to the area where they are required most. Because in one study at ICMR, very old study, it was found that zinc is associated with zinc deficiency, is associated with dwarfism, the growth of child. But giving zinc or replacing zinc was not fruitful. So what is your comment regarding this? By giving the vitamins, minerals, or fortifying them in our food, whether it is going to act in the positive way?
Actually, very good question, Dr. Bharma. One of my most favorite question. You know, when we take antioxidants, anything, it may be a botanical, it may be a vitamin, it may be an antioxidant or micronutrients. Now, what we did, we developed a technology how we enhance the bioavailability. You take a supplement and then it goes down your digestive tract and you flush it out with urine or feces. That will not help us. But if we can enhance the bioavailability, and that's the purpose I mentioned about the proteasome technology developed by our team, and we are doing a novel technology that is also mentioned in our paper. And as soon as we get the patent, I will share the patent with you. What it does, it enhances the bioavailability because you mix some nutrients, and if you mix it appropriately, you will get synergism. If it is not mixed appropriately, you will get antagonism. So, a synergistic combination and a technology, how to enhance the bioavailability so that you can take the requisite amount of nutrients in your body. And that is what is very important. And that's what we developed in our technology, in the proteasome technology, that which enhance the bioavailability several fold. And we already published it, but I will share with you when we come and meet in person, or maybe later on by email.
Thank you, Dr. Bagchi, because you have pointed out very well, it is the bioavailability which is important. Then only it will be effective, because we are consuming a lot of turmeric and saying that we will get curcumin. No, because whatever curcumin is present in turmeric, it will be wasted in our GI tract by our acid.
Yes.
And whatever left will be absorbed will be metabolized in the liver. So it had to be first bypass of liver, then only the effect of curcumin will be seen all over the body. So very strong,
exactly.
You advocated this, and bioavailability is most important thing. Thank you very much for that.
Yeah. Yes. Yeah. Bill, do you like to comment something? My colleague, my R&D colleague, Bill, do you like to comment something?
I was just
Dr.
going to.
Bharma?
Yeah, thank you. It is a privilege to be here. Thank you very much. Sorry, I'm not dressed like Dr. Bagchi. Also the ligand to which the mineral, in the case of minerals, the ligand to which the mineral is bound has a great influence on its absorbability and its usability, where it's going to be transported and how well it's going to be used. Its efficacy. If you have a mineral that's like in the case of zinc, like we like the form of zinc bound to L-methionine, we have shown that that has 6 times greater bioaccessibility and bioactivity than, for example, a zinc sulfate. So the ligand plays an extremely important part, and also zinc methionine doesn't require metallothionein as copper does to get absorbed. So again, the form of the mineral plays an important part in its ability to access specific tissues. And that would be one reason why, if it's a wrong form or a poor form, it won't show up in certain tissues where you hope to have it or get the result that you would want to have it achieve if the form is not right.
Thank you. Thank you. Excellent comment. And I think this will be beneficial for our attendees. So they can find it out very well, because you talked about zinc. And we all are using iron in that particular form, because we know that iron absorption again depends upon which type of iron we are using. We know that a ferrous sulfate is a good one, but ferrous sulfate has got a lot of side effects. So lot of the designer iron came into the market, but the problem is that the iron absorbed only at the second part of duodenum. So if you make it like a designer iron, it is not going to be absorbed in the whole of intestine. So your comment is very valid and very knowledgeable. Thank you. Thank you very much.
Thank you.
Thank
It's a
you very
pleasure.
much.
Yeah. One question I will like to put for Dr. Shipra Bharadwaj. We are using a lot of phytoestrogens just to treat the symptoms of menorrhagia and symptoms of PMS, symptoms of menstruation syndrome, and also the postmenopausal symptoms. So what to suggest, whether these phytoestrogen can produce toxic effects which you was talking about? They can have some effect on androgen as well.
Toxic effects are mainly due to the synthetic xenoestrogens, not on the natural parts. Basically, phytoestrogens are the natural xenoestrogens.
Yeah.
So the toxicity complexes and because since they are used in the therapeutic uses, so the toxicity effects are nil or negligible. The side effects may be, it depends upon the dose. But as far as synthetic synthetic xenoestrogens are concerned, they are mainly used in food as chemicals or the pesticides. So, and they are not decided according to the dose components. So that's where the toxicity effect of synthetic xenoestrogens are more than the phytoestrogens.
I think it is a very valid comment, because we all are using a lot of phytoestrogens to treat the postmenopausal symptoms. And I especially prefer them because the hormonal therapy, HRT, is not a very good option. We know that hormonal therapy may have more side effects rather than phytoestrogens. So I prefer them. That is why I was worried, because you have worked in ITRC and in that particular area. So I wanted to know whether you have got any side effects which are alarming by using phytoestrogens. Because some papers have indicated that even phytoestrogens can expose the ladies to breast cancer. That is why I'm asking that, because they are xenoestrogens. Natural one, I agree, but they are xenoestrogens, and all xenoestrogens can produce a lot of cancers, including breast cancer. A lot of work has been done on that, and we have found that breast cancer is one of the area where these estrogens are very, very important. Dr. Arun Kedia, your lecture was very nice, and you worked very hard to give all this. And in the coming time, we know that population is rising all over the world. Population is rising. One particular thing what I have in my mind is that before independence, India was having less food, but the quality of food was good. Today we have plenty of food, but the quantity has increased, but the quality has gone down. What is your comment regarding this?
So this has been very nicely summarized as something called as hidden hunger. So we have ample food, we are growing more obese, but the quality of nutrition definitely is less. And the reasons for this basically is the lifestyle that we have been taking. And there have been a lot of various unnatural foods that we have been using in our day-to-day life. And introduction of all these chemicals, especially as Shipra ma'am also spoke about the various ways in which pesticides enter into our body and how the xenoestrogens are harming us without letting us know what is the effect in the body, and we are getting exposed to them every day. I think that is, these are all these factors, and especially the stressors that we have in our day-to-day life. And as Dr. Bagchi sir has already summarized in his talk, that one of the commonest cause of chronic inflammation in our body is these stressors, the physical stressors, the emotional stressors, and all these lead to lesser bioavailability. I should say they cause different pathways of metabolism in the body, and all these lead to different quality or the bad quality of nutrition that we have in our day-to-day life. So I think improving the lifestyle would be one of the important concerns to tackle all the problems of overnutrition or the so-called obesity that we are seeing in our country. So despite availability of a large quantity of food, we still have hunger in our country. Another important aspect is the regulatory concerns, because we know that the government is not able to reach to the needy people, and whatever policies are formed, they are actually not very much practically used. So this could be another reason for why we have still malnutrition in our country.
So what you think that genetically modified food can be an answer to this all?
So it cannot be the solution to all the problems. Genetically modification could be answers to only a few problems of malnutrition. So we need to identify certain groups of people who are malnourished. So if you see in Chhattisgarh, we have an area called Bastar district, where we have a large tribal population which is very, very malnourished. So probably, we have to identify these groups of population in various corners of the country where adequate food does not reach, or where they have a very monotonous diet. So they eat rice all the 3 times of the day. They do not have exposure to multiple different types of nutrients. And probably even despite having a good quantity of a particular single diet, they may not have adequate nutrition. So we need to identify all these groups of population and target them with genetically modification of foods. But all the problems of malnutrition or overnutrition cannot be tackled through genetic modification.
I agree to you. That reminds me one study which was done by me when I was just a JR1R resident in the department. That study was done, and the comparison was made between the hostel inmates, female students, versus Bilaspuri laborers. Bilaspuri laborers were taking only the jaggery and bread, while the inmate of hostels were having the full meal. And surprisingly, only one parameter was studied: hemoglobin. Hemoglobin was found less in the hostel inmates of female students in comparison to the Bilaspuri laborers. And the reason explanation given was that they take plenty of bread and jaggery, which is enough to give a required iron. While the hostel inmates were having a lot of food fats, I will not eat this, I will not eat this, this is very harmful, and this is not tasty. So they were having less hemoglobin in comparison to Bilaspuri laborers. Another reason can be because they were working hard. So hypoxia inducing more erythropoietin, and that was responsible for the hemoglobin in the Bilaspuri laborers. So I agree to your point, and this is coming to my mind that over the years, we have more options, but the quality of food is dying. We have more delicious food, but less nutritious food. So that is hidden hunger, what you're talking about. Dr. Devasis has given us a very important clue regarding obesity. He was talking that the body had its own rhythm. It will come again. Whatever you do, whatever weight loss you achieve, it will come again. And he commented very nicely that fat is very difficult to dissolve. It is very, very immortal thing. Dr. Bakshi, last comment from you. You was talking about fat. Fat is immortal.
Yeah, exactly. So we have to trigger the function which can cause the fat metabolism. And that's what we are doing, so that these nutrients, they can go to the right cellular site and they can initiate the breakdown of the fat. And we can determine it two ways. One way is by measuring the genes, or the other way, these fats break down in urinary metabolites: formaldehyde, acetaldehyde, acetone, and malondialdehyde. Small amount, and that you can measure as a biomarker of fat oxidation. And we published that paper a couple of times. I will send you some of those papers also. So, the triggering the mechanism, that's what is very important. So if we give a good bioavailable supplement which triggers this process, then we will be successful. And we are leading to that way. And I will send you our proteasome technology video. You will see how it is enhancing the bioavailability of these nutrients. And don't forget grandma's technology, mixing turmeric with honey. And you have seen turmeric and bioperin, it helps. So there are a lot of benefits we have. So, we have to enhance the bioavailability and absorbability in the body. And that's why we are focusing for.
Thank you, Dr. Bakshi. So I think we have crossed the limit of time. And I'm very, very thankful to our Professor R.B. Singh, sir, who started the whole thing. And he was here to stimulate the thinking and the whole program. My governor, ACP, Dr. Anuj Maheshwari, and speakers, Dr. Devashish Bagchi, in the very early morning at US, he has joined us. So Dr. Maheshwari has come. So Dr. Bagchi for joining us very early in the morning from US. Dr. Shipra Bharadwaj, making all the trouble in just one day, she prepared all. And the good thing was that she was working in ITRC in the same area. So I think it was thought-provoking for her to present. And Dr. Arun Kedia. Arun Kedia is a marvelous speaker. He can speak on any topic given the time, and he makes his own adjustment. That is very nice. I'm very, very, I'm thinking that our listeners would have gone a lot of benefit from this particular seminar. Now it is over to Governor Dr. Anuj Maheshwari.
Thank you, Dr. Verma. And I must pass on my thanks to you that you have very nicely convened the program. You have given the concept of organizing this program. You have conceptualized and finally found the speakers, appropriate speakers for this program. I must pass on my thanks to respected Dr. R.B. Singh, who has been a very important person in this field, and he has been mentor of many of us, including me, Dr. Verma, and many others. So he has really given a very nice vision to this meeting of important topics and very nice speakers giving his expertise. And as much as I praise Dr. Shipra, that is less, because Dr. Shipra has done the fantastic job. She has prepared such a difficult topic in only 24 hours notice, when we found that our already fixed up speaker is not being able to come up. And this topic has been presented very nicely. Devashish Bagchi sir is already expert of his job. So we were expecting what he has already done. And I'm really grateful of him that I have had an opportunity to write some of the papers with him, and with good publication I'm sharing with him. Dr. Arun Kedia, again, he is a person who needs a lot of appreciation. Dr. Arun Kedia was speaking just like that. He is having excellent expertise in the topic, and I shall definitely be looking for his talk or his presentation in coming days with the ACP and other ACP programs. Dr. Debashish Bakshi, I think that you will also be available for American College of Physicians India chapter in future also. We shall be organizing our annual conference, and I will discuss the agenda of that so that we can have your talk in that kind of a meeting also. With these words, I conclude the program. Only the concluding message has been given by Professor Verma and Dr. R.V. Singh. I simply pay my gratefulness and thanks to all the participants, those who have attended, organizers, RX events, and the speakers and moderators, those who were there for a long and late hours of the day. Thank you.
Thank you, sir.
Thank you. Thank you.
Thank
Good
you.
morning.